Hello, everyone, and welcome. We'd like to get the presentation started now. I wanted to welcome everyone to this educational webinar titled Making Lung Cells from Human Pluripotent Stem Cells, Disease Modeling and Future Therapies, part of our webinar series in the Pluripotent Learning Lounge. My name is Simon Hilkov. I'm a product manager at STEMCELL Technologies, and I'll be your host and moderator for this webinar. It's now my pleasure to introduce you to our speakers today. Dr. Janet Rossant is a senior scientist and chief of research emeritus at the Hospital for Sick Children in Toronto, president and scientific director of the Gardner Foundation, and a professor at the University of Toronto in the Department of Molecular Genetics, Obstetrics, Gynecology, and Pediatrics. Janet is an internationally recognized developmental and stem cell biologist, exploring the origins of stem cells in the early embryo, including the discovery of the trophoblast stem cell and their applications to understanding and treating human disease. Dr. Amy Wong has obtained her doctorate from the University of Toronto, where she studied the role of bone marrow-derived stem cells in airway regeneration. She's currently a senior research associate in Janet's laboratory and recently has authored publications in Nature Biotech and Nature Protocols describing directed differentiation of hPSCs into mature airway epithelial cells. Janet and Amy are currently working on generating renewable sources of airway lung epithelial type cells for personalized drug screening in cystic fibrosis. So let me now hand you over to Janet to begin the presentation.
Good afternoon. It's a pleasure to be here. So I'm Janet Rossant, and along with me I have Amy Wong. I'm going to do the presentation, and between us we will address any questions that come up at the end. So today we're going to be talking about specific application of pluripotent stem cells and their differentiation towards cells that derive from the lung to be able to model lung disease and think about therapeutic implications. So Amy is going to run the slides. So just to begin and put us all in the same starting point, I think everybody is aware now, of course, of the power of pluripotent stem cells. First, the derivation of embryonic stem cells in the mouse, and then, of course, importantly in the human. But really, things have taken off with the discovery that you can take adult cells and transform them back into cells that resemble embryonic stem cells, the so-called induced pluripotent stem cells. And in this slide, this is just one of the many kinds of slides that shows you how you can go from fibroblasts, adding particular transcription factors, the Yamanaka factors in this case, and transform fibroblasts or other adult cells to pluripotent cells, iPS cells. The power of iPS cells really is that for the first time we have the ability to make pluripotent cells from everybody, every human being, and in particular patient-specific stem cells that you can take from people with different diseases and begin to model that disease in the petri dish. So it's really that application of pluripotent stem cells that we want to talk about today, the ability to take adult cells and transform them back from patients and model a disease in the petri dish, study the phenotype, and think about developing new therapies based on the cells in culture, their responses to drugs, or indeed, potentially in the future, developing new replacement cell therapies that are personalized to the donor of those cells. So the disease that we work on here at the Hospital for Sick Children that Amy and I are focused on is cystic fibrosis. So cystic fibrosis is really the commonest recessive genetic disorder. Its incidence in Canada is 1 in 2,500, 2,500 people. And around the world it varies, but it's still a very high frequency. And it's a devastating disease. It causes problems in many epithelial tissues, lung, gut, pancreas, etc. But really it is the lung problems that are life-threatening because patients are unable to clear mucus from their lungs, and patients always say it feels like drowning. They cannot clear their lung, they get lots of infections, and until recently it was really a disease that killed young people in their teenage years. We are now seeing extended survival for cystic fibrosis, but it's still, the morbidity is very high, and the final treatment really is only lung transplant, which in itself, of course, is not a long-term survival treatment. The defect in cystic fibrosis, the gene was cloned here many years ago, sick kids, and it's a defect, a single gene defect in the CFTR chloride channel. So we know what the gene is, but still, 25 years later, it's only recently that people have been developing drugs that are actually designed to help replace and repair and reactivate, if you like, the CFTR chloride channel. Those drugs are coming into play. They're being now used in the clinic to treat children with cystic fibrosis, but it's not still a perfect treatment, only works with certain mutations, and patients vary in their responses. So there's a lot of interest in being able to better model cystic fibrosis in the Petri dish, and in particular because the lung is the most severely affected tissue, to be able to take lung, to generate a renewable resource of lung cells from cystic fibrosis patients, to be able to do drug testing, drug screens, and eventually perhaps replacement cells for therapy. So just to summarize then, the idea, and something that we are undertaking here at the Hospital for Sick Children, is to take skin cells or other cells from CF patients, reprogram them by the IPS approach into personalized IPS cells, and then differentiate them into lung epithelium in the Petri dish that we can use to study the function or dysfunction of CFTR in different mutation backgrounds, develop therapeutics, and again, potentially transplantation. We are now here at Sick Kids generating large numbers of cell lines from different patients with the same mutation on different backgrounds, different mutations, trying to capture essentially the whole range of mutant phenotypes that are available in the cystic fibrosis population. It's a long-term goal of really developing personalized treatments. But to get there, and that's a long-term goal, to get there we really need to do what's shown on this slide, which is take those pluripotent cells and cause them to differentiate into lung cells. And how do we do that? Has really been our focus, and the focus of many labs in the last few years has been to take pluripotent embryonic stem cells and drive them towards lung epithelial differentiation. And clearly for any differentiated cell type or any tissue or any disease that you want to model in a dish, the goal is to be able to drive pluripotent cells down the differentiation pathways that they would normally undertake in the embryo and then into the adult themselves. So developmental biology is incredibly important, and essentially what we aim to do to generate lung cells is to replicate the events of differentiation and maturation that occur during lung development in vivo. This slide just summarizes what's in the literature currently. You can look at it later. I'm not going to go through it in great detail, just to point out that different groups have different goals when they're trying to derive lung epithelial cells. If you follow the cross-diagonal, do we have a pointer? Yes, we do. If you follow this diagonal, this is essentially the route that Amy has been taking to try to take ES cells through to epithelial cells, and in particular, proximal airway epithelial cells. We're focused on that because that's the area that is most severely affected in CF, and where ciliated epithelial cells expressed in CFTR are the cells that are most severely affected. So that's been our goal. Other groups have taken different side steps here, trying to drive cells towards, for example, respiratory epithelial, the alveolar cells that actually are the gas exchange. So there's a lot of interest, obviously, in other diseases in being able to model and mimic this process. So you have to take a different pathway. So if you look at these different pathways, you'll see that essentially in each case, what's happening is people are taking different routes we know from development and taking the cells down the different routes. So I'm going to focus, however, on the development of the proximal epithelial cells and the protocol that Amy has developed. So essentially what we're doing here is replicating what goes on in development. First of all, if you want to take pluripotent cells and you want to make them into lung epithelium is a definitive endoderm derivative. So the first thing that you do is drive your embryonic stem cells or your pluripotent cells towards definitive endoderm. And the original protocol we use for this uses what's known from the literature to be important. Activin A, which would in the normal embryo would actually be nodal signaling, but it's replaced by activin usually, it's cheaper, and Wnt3A. So first of all, you push the cells towards definitive endoderm, away from your ectoderm and mesoderm. Then you have to begin to pattern the definitive endoderm, which of course is going to form the whole of the gut, and we want to make the cells of the lungs, so they arise from the ventral foregut, anterior ventral foregut. So adding FGF2 and sonic hedgehog pushes the cells away from caudal fates, liver and gut, towards anterior fate and lung. And then you're taking cells that are pushed towards lung progenitors and starting to specify lung fate and try to move them away from the other foregut endoderm fates with another selection of growth factors. And then finally, as we move forward, this is where we know, I would say, much less about the phases and the detailed factors that are involved. We can get lung progenitors. We know from mouse mutations, zebrafish models, no, not zebrafish, they don't have lungs, frog models, other systems. We know the pathways that lead on to the sort of development of the endoderm and the foregut. And from the mouse system, we have a pretty good understanding of those that make lung progenitors. But what we don't understand as well is how to take those lung cells and transform them into mature lung epithelium. So at this stage, what we do is we take the cells and we put them in a mixed medium that is known to support lung epithelial cells, and that's the Beger medium. And then finally, because we want to make a polarized epithelium that really mimics the way the proximal epithelium is meeting the air-liquid interface in the lung, we move those cells into an air-liquid interface culture, so that the epithelial cells become polarized and start to become ciliated and functional epithelial cells. That's the overall goal. There are various other protocols in the literature and variations on this theme with slight differences along the way. This is what works for us and has worked very consistently. So when I say it works consistently, what do I mean? What do we get at the end of this? Essentially, what we can get is very nice, tight junction coupled, polarized epithelial cells that are ciliated, which essentially have the markers of large airway epithelia. And if you focus in on beautiful areas like this, you can get really large patches of these epithelial cells. However, if you take the whole culture, this is not a completely uniform epithelial layer. We are not, in this protocol, we are not getting a completely homogeneous differentiation. And you can tell that because if you look at the bottom, we have the FACS profiles. Epithelial cells, about 50% of the culture. CFTR also around the 50%. A subset of these cells express P63, which would make them basal cells. And again, the FOXJ1 is expressed in the epithelial cells. So we're making ciliated epithelial cells expressing CFTR, but it's not a completely homogeneous culture. However, there are big enough patches that we can actually see that these are functional epithelial cells that can transport chloride. And if you just look for CFTR expression in these patches, which you see here, we're not showing a completely homogeneous set of cells. We see that within the patches that are tight junction coupled, we see nice polarized, apical polarized expression of CFTR in those epithelial cells. And in the cultures that have large enough areas of junctionally coupled epithelium, we can actually measure chloride conductance. And this is work that we do in collaboration with Christine Baer's lab here at the Hospital for Sick Children, who are really experts in studying the biochemical function of CFTR. And here what you see in the red is the response to forskolin from wild-type ES cells or iPS. The green line is actually an iPS cell line derived from a CF patient, which does not respond, and then the DMSO is just the control and the wild type. So we can measure the chloride conductance, and we can see the difference between the mutant and the wild type cells. So this says we have an assay, and we're on our way. But I told you it wasn't completely homogeneous, so what else is going on in those cultures? Well, mostly we think what's going on, the additional cells are mostly endoderm cells, so we're pushing cells down the right pathway, but we're still getting somewhat contamination with other foregut endoderm-derived cells, particularly thyroid, pancreas, liver cells. So small percentages, but we're not then getting pure lung. So clearly, this is a kind of problem you see with a number of differentiation protocols from pluripotent cells, not just in lung, but in other cell types as well. The ability to get an absolutely homogeneous population is tricky, and to get mature cells that are fully functional is tricky. We think we have good maturation of these cells, at least a good enough maturation to be able to model and study CFTR And what do we do to try to improve the differentiation, the maturation, and the homeostasis? And that's what we're doing right now. And also, of course, sorting the cells at different stages. I'll come to that in a minute. The first thing we've done to improve the differentiation protocol, we published in our Nature Protocols article last year, and that was to simply improve the first stage. So if you want to have cells that are derived from a specific germ layer, the first thing you want to do is to make sure that all your cells go down that particular pathway and differentiate into that particular germ layer. And we have found that replacing activin A and Wnt3A with the definitive endoderm kit that Stem Cell Technologies has generated, which has a number of different growth factors, I'm sure they won't tell me what they are, but, but that particular modified and reproducible medium in our hands has really helped the differentiation protocol. We get a much better purity of endoderm, as we'll see in the next slide. And then if we just take the cells after that step and take them through an unmodification of the, no real modification of the next steps, we improve our overall differentiation, and I'll show you that. So here now, the original protocol, if we just go to stage 2 and look for the embryonic lung progenitors, if we do FACS sorting for FOXA2, which is a definitive endoderm marker, we're getting about 80%, and about half of them are expressing NKX2.1. If we do the replacement protocol, now if we look at FOXA2, which is our definitive endoderm marker, almost everybody has gone and made definitive endoderm. And now when you look at the lung progenitor, instead of 55, we've gone to 75%. Still not everybody is lung, but again, this is early in the protocol. But if you then take all those cells through the rest of the protocol right to the end, the point at which they're growing in the air-liquid interface, and look at them functionally, now we see a much more uniform production of our epithelial sheets, not shown here, but if you just look for the overall expression of CFTR by protein in those cultures, in those cells, you get a whole load more particularly mature CFTR protein. So we're getting better uniform epithelial cultures, better maturation, better CFTR expression, and as you see on the right, all of those cell lines are responsive to forskolin. So we now have a system that is good enough, we believe, to start using in drug-type assays. So how would we use these cells? This is really the work that we're doing in collaboration with the CS Center here in Toronto. Christine Baer, the biochemist in this group, Felix Rathjen is the clinical head of the CF clinic, who has patients who are being treated with the newly developed drugs against the CF gene. And what we're trying to do is to provide the clinic with some information on which patients are likely to respond to which drug. So on the top you see the sort of proof of principle when we first did this differentiation protocol without the full organized epithelial sheets, that we could see some response to the drugs. So what we see here on the top, you see the normal wild-type cells with CFTR expression on the membrane. In the middle, this is what happens when you make lung epithelial cells from CF patients, iPS cells. They make epithelial sheets, but there's no CFTR on the surface. This is the most common CF mutation. It's the delta F508 mutation, which is an in-frame small deletion. And that CF protein is unable to be processed to the membrane. And you can see it in little dots inside the cells because in the ER. If it gets to the surface, it can be somewhat functional, although it still needs to be modified further. And on the right, you see treating those cells with one of the developing drugs that people are using to repackage this protein. You can see now that we can find CFTR protein on the cell membranes. In this assay, we can't see function, and again, not enough protein and not enough functional protein still. On the bottom, we see a further step that Christine's lab is now taking. She's taking our epithelial cells and being able to transfer this into a 96-well plate, a 96-well culture system in which each of those little wells contains our air-liquid interface polarized epithelium. And in this case, what they're doing is treating us with two drugs, one to repackage the protein and one to potentiate its function. And on the right, we see a sort of first example where we're taking two different patients, both with the in-frame deletion, and treating them with the combination, and we see a different response. So same mutation, different genetic background, one patient responds well, and the other patient doesn't respond at all. Now what we're doing, of course, is comparing this to how the patients in the clinic are responding to see whether this is a good test. This is the kind of goal that many people are trying to achieve in this disease and in any other disease, using our cells, using the genetic variation in iPS cells to really be able to predict patient response to drugs. So we still don't think we're perfect. We're not perfect. It's still not absolutely homogeneous. So we are continuing to improve the protocols in an ongoing process. We're also trying to think about other assays. So going all the way through to get differentiated lung epithelial cells takes about 45 days, and it's expensive. There's a lot of cells and a lot of reagents along the way. We think that having, certainly for the final validation of any drug responses, we really do want to have mature lung epithelial cells. But for a first-line, relatively high throughput screen, we probably need a simpler system. So we are trying to think about moving how far back in the protocol can we go and still isolate progenitors and develop perhaps organoids that would still express the FTR, be useful for drug screens, but would be cheaper and easier to generate. So we're working on that, and we'll show you a little of the work in progress here. This is certainly not complete work at this time. But the idea here is to see whether we can develop a sphere or organoid assay from the developing lung epithelial cells differentiating from the ES cells or iPS cells. And this is a sort of following on the original sort of bronchosphere assay that was developed by Bridget Hogan's lab, where they were able to show that proximal airway epithelium can make what they call bronchospheres. And so the question is, can we make bronchospheres during ES differentiation in culture? There have been some recent papers suggesting that it is possible to get organized, lung-type organoids, so we are trying to perfect this along the way. So what can we find? So if we transfer the cells that are growing in flat cultures into a suspension culture with the right factors, we can't get spheres from definitive endodome. We can't get spheres from the next stage, making lung progenitors, but we can generate spheres from the stage 3 and stage 4 immature lung cell differentiation stages of our protocol, and they look quite pretty. The next slide shows that what we would like to be able to do is during that stage where if we just take the whole culture, we can get spheres of various sizes and various attributes. This is the stage at which we'd really like to be able to isolate progenitors and be able to generate hopefully single cell derived spheroids that we can really develop into a reproducible assay. So we know quite a bit about gene expression from a number of different studies going on during this differentiation. We've been focused on asking, are there cell surface markers that we might be able to select a subset of the developing lung progenitors that would be the ones that generate the spheres. So that would be a way of us purifying our cells and also developing a really reproducible assay. So this is very much in progress. So I'll just say this is where we're at today in the next slide. So Amy has used a number of different antibodies to the cell surface markers that we know come up during this differentiation process. And at this stage, I would say that EpCAM, which we know is a general marker of lung epithelial cells, is still by far and away the best marker that we have to enrich for sphere-forming cells. So by using this, we can get rid of non-epithelial cells, and we have not, to date, been able to use any other, at least single marker, that reproducibly enriches the sphere-forming cells against other cell types. And actually, this is consistent with other work from my lab, from Melanie Bilodeau, who was a postdoc in the lab, working in the mouse, where she was trying to isolate proximal progenitors during fetal development, and again found that EpCAM was the best marker, and that we have not yet been able to identify a specific subset of EpCAM cells that are the spheroid progenitors. Work in progress, but that's sort of where we're at today, leaving open the question of whether there is such a thing as a restricted progenitor at that stage. Anyway, what do they look like? This just shows you the pictures that we have to date, and just showing that we produce these very nice spheroids, and that they do express lung epithelial markers, and importantly that they express CFTR on the luminal surface. So this is now, we are doing further analysis of these, further characterization, and we do hope that this can be moved into a more of a first-line assay for drug development and drug testing, using our epithelial cultures as the final differentiation assay. This is just to show that the sphere-forming cells and the spheroids do actually express the FDR, and again that we can see a response to forskolin. So we believe that this is an assay that should work. So that's the kind of approach we've been using to generate from patient iPS cells, lung cells, particularly proximal airway epithelial cells, that we can use to study the disease, study CFTR function, and develop high throughput and more complex assays for CFTR function to test known drugs, to test patient-specific responses, and potentially to develop new drug screens. Could this be used in the long run as a replacement therapy? What about using lung epithelial cells derived from iPS cells or ES cells to replace damaged lung tissue in CF, or indeed in other lung diseases or lung injury? There is certainly interest in this approach in many different areas, but it's tricky in many ways. And certainly the kind of differentiation we just showed you today, it's difficult, it's slow, it's expensive. And still, if we wanted to take our cultures and put them back into patients as it stands, I don't believe we would be able to produce a safe product, because we do not have a homogeneous product. We do not necessarily have full maturation of all the lung epithelial cell types. We're focused on ciliated cells, so we're quite happy. But of course, the lung epithelium is a complex epithelium with many different cell types. And if you really wanted to have a replacement system, you'd really want to be able to be sure that you could generate all cell types needed. You don't, of course, necessarily replacing the lung epithelium itself may not be enough. You have to reconstitute the whole epithelial niche when you have complete damage, and stromal cells, endothelial cells need to be incorporated. And people are looking at that and generating more complex organoids, for example, which may lead down that pathway. So I believe that lung regeneration is one of the more challenging applications of iPS technology, iPS technology, but it's actually one of the more rewarding applications when it comes to studying disease in the petri dish. How could we, are there ways that we can see going forward with regeneration? I talked about organoids, making better organoids. There's some interesting observations, however, that suggest that in the lung and perhaps other tissues, that our focus in pluripotent differentiation on making sure that we have everything in the petri dish developed into the most mature state before we put it back into the preclinical model or into a patient may not be entirely necessary. That if we can get cells into the correct environment, the local environment may help mature and differentiate those cells. And I just want to point out this recent paper from Martin Post's lab here at Sick Kids. We were collaborators on this project, in which they were taking from ES cells early definitive endoderm progenitors or early lung progenitors, so that would be sort of stage 1 and stage 2 in our differentiation protocol, and putting them directly, not into the lung, but into acellular lung scaffolds in culture. And in that situation, those epithelial cells can repopulate the matrix and start to differentiate. And you see here that they were able to see differentiation expressing NKX 2.1, and if they continue those cultures, they were actually able to see ciliated cells and other apparently differentiated lung cell types from early progenitors. So again, suggesting that, although I was just being rather negative about lung as a target, that it may be possible if we can develop better preclinical models, not just lung scaffolds in culture, but preclinical models to study this in animal models, that we may be able to find ways where the lung tissue, if not completely damaged itself, may help repair. So we can provide cells that the lung itself will mature. So this is really, again, one of these sort of overview slides that you can take away and have a look at afterwards. This is just my thoughts on sort of where we are today in terms of the different routes that one might be taking towards getting renewable lung tissue. Renewable lung tissue for studying disease, or renewable lung tissue potentially in the long run for putting back into patients to repair tissues. So again, going down the middle here, we see the differentiation pathway all the way through to epithelium. But along the way, if you take out, progenitors and put them into a lung scaffold at different stages, can you actually get better differentiation? In the long run, could we do the same thing and take these progenitors and put them back into the lung in a preclinical model and look at maturation and repair? What is going on here? How far can you go down the pathway from a cell in culture and using the lung scaffold or the lung niche to drive its differentiation? Can we get renewable progenitors out at different stages? At what stage? Can we get early progenitors? Can we get renewable progenitors from the proximal epithelium? Can we take those renewable progenitors, combine them with lung stroma and get better lung organoids to study disease, or perhaps better understand how to repair tissue? Can we do what people have done in other cell types, and that is avoid this pluripotency route altogether and do direct transdifferentiation? Can we take fibroblasts and turn them directly into epithelial progenitors, which perhaps themselves could become renewable progenitors, perhaps could go on and differentiate, perhaps could be combined to make organoids. So I believe there's a lot of different pathways here and still a lot of questions that we don't have all the answers to. And that's where I'm going to stop today. Really, almost all the work I talked to you about today, I should have let Amy talk about, who's sitting next to me, because this is really, she's been the driver of this work in the lab. We have very great collaborators here at the Hospital for Sick Children. Cystic fibrosis is a major clinical area here at Sick Kids, and of course therefore a major research area. James Ellis is involved in the early generation of CF iPS cells, Christine Baer, the biochemistry, people in her lab, Felix for the clinical work, Martin Post and Sharia Shojai for the lung scaffolds, and then Jody Garner here in the lab, who really runs our ESL facility. And funding, of course, from Sick Kids, Cystic Fibrosis and CHR. Thank you.
So as we get a couple more questions in, I may just take the opportunity to ask the first one that's of interest to us at STEMCELL. I was just wondering if you could comment on the role that you see of the standardized reagents that your lab is using, like MTeser or the STEMDiff DE kit, in terms of the consistency you see across cell lines or across experiments or even across labs as protocols get shared.
Yeah, so first, certainly the STEMDiff definitive endoderm kit has really helped us in terms of getting higher efficiencies for the endoderm progenitor cells that we eventually see and eventually downstream proximal area epithelial cells. But what's really good about that kit is that for all the lines that we've used, both different ES lines as well as IPS lines, it's consistently given us higher percentages of definitive endoderm, which really helps with the differentiation downstream.
And we have transferred the protocol to other labs. We've had people come and visit us here to learn the protocol. And again, I think we'd be able to transfer it from lab to lab and from cell line to cell line. But certainly having that early stages being robust and reproducible helps a lot.
Great. Okay, so one of the questions here is from, have you guys used the Ussing chamber to study the CFTR current and drug response using ALI culture?
Yeah, so we have attempted to use that Ussing chamber for the earlier studies, but as Janet mentioned, the differentiation was not homogeneous, and unfortunately for the Ussing chamber, you do need a homogeneous population of tight junctioned epithelium to have good measurement, and in 2012 we didn't have that kind of cultures available to do that kind of Ussing chamber studies. And then for our nature protocols, although we have improved the differentiation, as Janet did mention, we still see some contamination, and so the Ussing chamber studies were always variable. So right now our focus is to improve the differentiation by isolating those progenitor cells to get a more homogeneous population of cells to do the Ussing chamber studies and other types of CFTR functional studies.
So we do have there are other kinds of assays that the lab is using that are easier to work with and that require the assaying chamber.
So you had some interesting stuff about the organoids in there. So one of the questions is about your organoids. They don't appear to be positive for TTF1 completely throughout, and instead it seems like a few of the organoids have TTF1 positivity. So we're wondering if you have been able to see if the TTF1 positive organoids are swelling or responding to forskolin, or are the TTF1 negative cells responding?
So that's a good observation, and that is the next step. So as Janet mentioned, the marker that we use to isolate the cells to generate the organoid is CD326 or EpCAM, and it has consistently given us organoids. However, they're not all TTF1 positive organoids, so our next step is to follow through with the TTF1 positive organoids to see if they do swell in response to forskolin.
Okay. On the topic of the organoids, we were wondering what medium you've used for the organoid culture or for the ALI culture for that matter.
At this point, that's our secret.
We cannot disclose the organoid media. However, the air liquid interface media is published, so that is from Lonza, and it's the BALI kit. Okay.
Another of the questions was just about how are you able to expand the epithelial cells that you're generating? Askers had difficulty with expanding primary cells and maintaining the morphology. Just if you've had success with that.
So we haven't really tried to expand the cells over serial passages at the different stages. It's something that we are currently looking into, not only expanding them but freeze thawing them for other purposes. But it's something our lab is looking into right now.
It is tricky, there's no question. So by the time you get full differentiation, it's unlikely that you're going to get a whole load of expansion of those cells. So really, to get a lot of cells, you have to start with a lot of cells, and that's the other reason why this is quite an expensive protocol.
Speaking of the protocol, people are wondering about the matrix that you're using. It sounded like at first maybe you were using collagen 4, but then in the Nature protocols you're transferring to Matrigel. I was just wondering if you could comment on the effect of that change in proteins.
Yeah, so the original protocol the entire differentiation was on collagen 4, but then we wanted to expand more cells for air liquid interfaces, so we kept the differentiation in the petri dish on Matrigel coated plates to get that expansion going, and then eventually at different stages of the differentiation we can then put them on their air liquid interface cultures that are coated with collagen 4. So they eventually still need collagen 4 for the maturation, but you can keep them in Matrigel coated cultures earlier.
Are you able to freeze the cells at any point during the differentiation?
So like I mentioned, it's something we're working on right now to see if we can freeze thaw the cells.
I don't know, maybe a broader question. You hinted a little bit about the ability to use these IPS models to identify the right drugs for people that may have CFTR, kind of in a direction towards personalized medicine. So I was just wondering how you feel about the potential of that and how far away you think this field is from getting to something like that where it could become a routine part of care.
Well, I think there's a lot of push actually in a number of places around the world. So in Europe, they're already talking about using gut organoids as an assay for what patients to treat. And certainly here, there are a number of different assays you can think about doing from patients. From iPS cells, we can generate lung, we can generate cholangiocytes, which also form little cysts, we can generate liver cells, we can generate all sorts of cells, and you can generate primary cultures, primary epithelial cultures, and gut cultures from the same patient. So, this is an area where there's a lot of interest in trying to work out what is going to be the most robust, simple, and most consistent readout of patient responses. There are now drugs in trials, so patients are being treated with different combinations, and those patients have different mutant backgrounds. So and the drugs are extraordinarily expensive, and not every patient responds, as I say, even with the same mutation. So there is quite a high push from our clinical colleagues to come up with some kind of way of telling ahead of time who's going to respond. Who's going to respond well, who's not going to respond at all, and who's going to respond badly. We are really pushing this quite hard here.
Right.
We have funding to build this up.
Yeah, it's definitely very exciting. Another interesting question about the, we're doing a lot of work to try to make the most mature and functional cells we can. It's, we're just wondering, is it possible, do you think that the cell type that will do the most regeneration of sick tissue, is that going to be the really mature cell type, or possibly some kind of progenitor, or I guess maybe a little too early, but can you speculate?
Yeah, I mean, I think that's an important question, and it's sort of a little bit where I was going at the end with the concept that actually the progenitors themselves, if put in the right environment, may be the best cells to respond to the environment and differentiate to the right cell types. And I do believe that is probably the case. I think the epithelial, it depends on the damage and the disease. So sometimes there is, lung tissue damage that just takes out the epithelium, which will repair itself to some degree. So if you could just get some epithelial cells that you could put back and kind of tide it over until its own cells can build up with it, you may be able to take fairly differentiated cells and use them. So it's just kind of like what people do with the skin, where they make skin epithelial cells for burn victims. I think you could think about making lung epithelial cells for a short-term repair for lung damage. But if you want to regenerate the whole tissue, particularly I think if you want to regenerate the respiratory surface, you probably are going to need progenitors that somehow respond to the local environment.
Another question is, have you looked at counter-regulatory ion channels in your system, apart from CFTR, in the cystic fibrosis iPSC?
No, we have not.
I
don't think Christine has. She is really just focused on CFTR right now.
Right, okay. So, apart from collagen, are you looking, or have you looked at other substrates? Just, when you think about the right environment, what types of factors are you looking at?
Yeah, so we have not looked at other extracellular matrix proteins and their involvement in the in the maturation of the differentiated cells. Our goal now is to look at the decellularized lung itself. Of course, the decellularized lung has a whole bunch of different extracellular matrix proteins. We're looking at the role of decellularized lung matrix and how that matrix can play a role in the directed differentiation of those progenitor cells.
Okay. I think we're almost getting to the end here. Maybe just one or two more questions. So there's been a couple of papers published recently using CPM to sort proximal lung progenitors. Just wondering if you tried that and if it's worked well for you.
It was on our list.
Yeah. Yeah, it was on our list. And as Janet said, it was pretty inconsistent in terms of being able to generate organoids from the CPM sorted cells.
That is to say, it wasn't differentiated. I mean, it was the positive and negative were the same.
Were the same, yeah.
Okay. One last question is just, have you had any spontaneous formation of goblet or mucin from these cells?
Yeah, so we do see a lot of mucus production actually in the air liquid interface cultures. Quite interesting because in patients that we know have severe lung disease with a lot of mucus production, their hPSC-derived cells also make a lot of mucus. So we do see a nice correlation. So we do see a lot of goblet cell differentiation in our differentiation, and it varies from patient to patient.
And maybe just to follow up, I guess, so have you been able to show primary cilia formation on the proximal epithelial cells?
Primary cilia or?
Yeah, like using any cilia markers or
yeah, so we have stained for some beta tubulin or I don't remember what we wrote on the paper, but we did see ciliated cells in our differentiated cultures.
Okay. I guess that's going to be all the time that we have for today. I really wanted to thank everyone for attending, and especially thank you, Amy and Janet, for your time and a really excellent and interesting presentation.